All right, last week's video, I made this claim that they were able to develop a cell over 200 million, it took over 200 million man hours. Now, I have to confess that I was quoting from memory and I couldn't find the article, so I do not know exactly how many hours it took. And I'm going to say people hours in case you thought only men worked on it, because I'm pretty sure women worked on it too. But the far more important impression I'm worried about is this impression that maybe you got from what I said that somebody actually made a synthetic cell. Craig Venter claimed to have made a synthetic cell using 4 bottles of chemicals, one containing A amino acid, one containing G amino acid, C, and T. If you know high school biology, you know how that works. But first, these amino acids came from sugar or soya, which comes from plants. Secondly, they put together these short strings of amino acids, not more than 100, and then they had to use complicated organic chemistry to get them to stick together. Once they got them stuck together in longer pieces, then they had to stick them in yeast cells and let the yeast cells put these longer strings together. When they got that done, then they once again put them in yeast cells and got them to actually form a string of DNA. Once they had this string of DNA, they then took DNA out of one cell and put the synthetic DNA in, and voila, they've made a cell. So let's poke a little fun at science. You call that making a cell? But the amino acids that you used came from plants. We can't easily synthesize amino acids ourselves. We have to get it from pre-existing organic life. Secondly, the organic compounds you use to put the amino acids together again came from already living systems. The long chains that you needed to put together in order to make your DNA came from the yeast. And when it came to actually making your DNA, you made it in the yeast too. Then you took an already existing cell and you put your synthetic DNA in there and you claimed that you've created a living cell. That's kind of a lot like copying and pasting Hamlet and calling yourself a playwright. But all that aside, where did they get the code? How did they know what order to put it in? Well, just like in my previous statement, what they did was they took the pre-existing organism and merely copied the AGCT code and put it into the new synthetic cell. So that is a long ways from making life. Just poking a little fun at science.